This is going to be a very interesting and complicated video, and I'm not sure how I'm going to put it off, but I'm going to give it a goddamn go anyway. I've been interested in science my entire life, and so this video is on the biosynthesis metabolism of cholesterol that is happening in pretty much every single cell of your body, because cholesterol is needed to make your cells fluid, it's needed to produce steroid hormones like testosterone, it's needed to produce bile acid, and it's a very interesting thing that I sort of went down the rabbit hole on and thought that this could be a potential thing that I do end up making a video on. And if they are wrong, if I did mention something wrong, please do tell me in the comments, because I don't want to spread misinformation on the internet. That would not be something that I want to do whatsoever. So yeah, and so here it is. I'm going to try to explain the complete biosynthesis of cholesterol, and hopefully it doesn't take me over an hour, because that will be an incredibly tough task that I've got to sort of edit and make it sort of interesting so that it's easy to explain visually in once I actually record this. And I have the steps of the metabolism of cholesterol right here in front of me, and there's probably around 50 or so reactions. I'm not going to include all of these chemical reactions in my video, but I'm going to try to cover as many as possible so that it sort of makes a little bit more sense than just going from step 1 to step 5, which is this chemical, and then you end up with cholesterol after this amount of time or whatever. And the first step in the biosynthesis of cholesterol is 2 acetyl-CoAs combining to make 1 aceto-acetyl-CoAs. Now, CoA stands for coenzyme A, and acetyl is actually something similar to what you find in vinegar. You may know vinegar is quite an acidic thing. Ethanoic acid is the acid that's actually found in vinegar, but many Americans call ethanoic acid acetic acid, and so acetyl is a specific part of ethanoic acid, just without a certain part of ethanoic acid, acetic acid, whatever you want to personally call it. And then after you get aceto-acetyl-CoA, you end up taking that aceto-acetyl coenzyme A, adding it to another acetyl group, and then once you add in HMG coenzyme A synthase, which is an enzyme, and enzymes are a thing that often catalyze chemical reactions, you end up with HMG coenzyme A, and that's a very complicated chemical, and I don't quite know the name of it. I think it's something like hydroxymethylglutaryl, and it's got a whole bunch of numbers in it if you sort of consider the systematic names, but yeah, I'm not going to go into that because that is a very complicated thing. And then once you get HMG coenzyme A, you take that HMG coenzyme A and you combine it with 2 NADPH, which are enzyme little chemical things that are produced by a certain bacteria called E. coli. Then you take the enzyme HMG CoA reductase, and then you get mevalonic acid. That's how you pronounce it. And this step right here is the rate controlling step. The rate that this reaction goes is the rate that it's hard to explain it. The rate that this reaction ends up going is the rate that the cholesterol is, once the end of the biosynthesis comes, is produced. Because when cholesterol is produced right at the end of this biosynthesis, cholesterol ends up being something that's needed to make the enzyme HMG reductase that we talked about earlier. I'm pretty sure, I might be wrong, please call me out if that is wrong, but I'm pretty sure that is how this step is controlled. And then once you get mevalonic acid, that's when things start to get a little bit more interesting. There's a chemical known as adenosine triphosphate, which reacts with the mevalonic acid, and that ends up, the triphosphate of the adenosine triphosphate ends up, there's a phosphate group which is PO4, I'm pretty sure, if you try to answer that chemically, ends up being added on to mevalonic acid, and this reaction happens a few times until you get, and the first of these reactions is when the mevalonate reacts with the adenosine triphosphate, as we talked about earlier, and this turns into 5-phosphomevalonate, as well as an ADP molecule, which stands for adenosine diphosphate, and this is because one of the phosphate groups is used up to make the phosphomevalonate molecule. And mevalonate 5-phosphotransferase is the enzyme that is used to catalyze this reaction, and after that, then you have 5-phosphomevalonate and another adenosine triphosphate molecule reacting to form 5-pyrophosphomevalonate and again another adenosine diphosphate molecule, because, as I said, it's added another single phosphate molecule on, and there's an adenosine diphosphate molecule that ends up not being used. And the enzyme that catalyzes this reaction is phosphomevalonate kinase. And once you have 5-pyrophosphomevalonate, then you react that with another adenosine triphosphate molecule, and then that will turn into a very, very, very complicated molecule known as 3-phospho-5-pyrophosphomevalonate. It's quite a mouthful, and again, like the last time, you end up getting an adenosine diphosphate molecule on the side because it's not needed. And the enzyme that is used to catalyze this reaction is known as kinase, and kinase is like a specific type of enzyme that binds proteins, I think it's to isoprenes, but that may be wrong. Please correct me in case that is wrong. And then once you've got this very complicated molecule, 3-phospho-5-pyrophosphomevalonate, this is the reaction that then turns it into isopentenyl pyrophosphate. And you don't need an adenosine triphosphate to do this reaction. It simply is turned into isopentenyl pyrophosphate by the enzyme pyrophosphomevalonate decarboxylase, and the wastage of this reaction just ends up being CO2 and a phosphate group, and then you've got isopentenyl pyrophosphate. And that's it's quite similar to mevalonic acid, just with like a few phosphate groups on the end, and you end up needing 6 of these isopentenyl pyrophosphate, to end up getting to the next step. And the next step contains a few reactions. You end up going from isopentenyl pyrophosphate to dimethylallyl pyrophosphate, and this switches the double bond, and there'll be images on screen which should help explain it, but it switches the double bond, and then you get that dimethylallyl pyrophosphate and add it to a separate isopentenyl pyrophosphate molecule, and then that will turn into geranyl pyrophosphate. And this reaction is catalyzed by the enzyme cis-prenyl transferase, and 2 phosphate groups end up becoming waste. And this geranyl pyrophosphate molecule, which contains 2 isoprenes, you then add another separate isopentenyl pyrophosphate molecule to the geranyl pyrophosphate. And again, this reaction is catalyzed by cis-prenyl transferase, and this will end up turning into farnesyl pyrophosphate. Now, again, 2 phosphate groups end up coming on the side as waste, and you need 2 farnesyl pyrophosphate molecules which contain 3 isoprenes, as well as 1 NADPH molecule, as well as the enzyme squalene synthase, which catalyzes this reaction to get squalene. Now, squalene is a very, very long, complicated molecule. It's a hydrocarbon. It's said to be quite good for your skin. And while it may seem quite long and look absolutely nothing like cholesterol, if you sort of shape the image and fold the bonds in slightly different directions, you'll realize that it does actually look quite a lot like cholesterol. You realize there's like a little bit where you can sort of see the sort of outlines of the rings forming. And then you take that squalene and you epoxidate it, and this is done by the enzyme squalene monooxygenase, which can also be known as squalene epoxidase, but on the research that I've done, I've found that it's called squalene monooxygenase. And you also need an NADPH molecule, which, as said before, is produced by the E. coli bacteria, as well as an oxygen molecule. And this will form squalene 2,3 epoxide, as well as some water as wastage and an NADP molecule. And whenever NADPH is involved in any of the chemical reactions that I've talked about in this video, NADP then normally ends up coming out as waste on the side, simply because the reaction only often needs the H part of the NADPH molecule in the first place. And then once you have the squalene 2,3 epoxide with a little sort of triangular oxygen which is bonded to 2 different carbons, and then you use the enzyme 2,3-epoxysqualene cyclase. All this stuff has incredibly complicated names, but this is more commonly known as cyclase, and this will put the bonds together so you can actually form the rings that you see in molecules like cholesterol. And then boom, you have lanosterol, and that is a molecule that actually looks very, very much like cholesterol. And the reason that you sort of had to do the initial reaction first with the squalene monooxygenase is because lanosterol has that little OH tail at the end, and that's why the oxygen that was added on in the last reaction was needed in the first place. But while lanosterol looks very, very similar to cholesterol, there are still 18 reaction steps that still need to be done before you end up getting cholesterol. And I'm not going to go through all 18 because I couldn't actually find all 18 reaction steps in my research, and I'm going to go through as many as humanly possible that I could find while doing my research. So you've got lanosterol, and there are a few paths that you can take to then get cholesterol, because if you look at the differences between lanosterol and cholesterol, you'll see that lanosterol has 3 methyl groups that are not supposed to be there. The double bond on the tail isn't supposed to be there, and the double bond inside the sort of rings is in a different place in cholesterol. They are the 3 changes that need to be made from lanosterol to cholesterol, and unbelievably that does end up taking 18 different reaction steps. Now, the reaction that gets the double bond off the tail can happen at any time whatsoever in the sort of reaction process from cholesterol. It can happen either at the start or at the end, and it doesn't really matter. But the other ones, I don't really know if they have to take place at a certain point in time, but I'm going to start with a certain pathway where you end up getting rid of the methyl groups first. And so you end up taking your lanosterol, and there are no enzymes that catalyze this particular reaction, and you add an NADPH molecule to it as well as an oxygen molecule, and that ends up turning into 14-desmethyl lanosterol, and this is the reaction that ends up taking away the tail that is right at the top of lanosterol. It's the methyl group that is on its own and is the one that isn't actually present in cholesterol. And in this reaction, there seems to be a little bit of wastage in the form of an HQ thing, and this is 2 hydrogens, 1 carbon, and 2 oxygens, and I'm not sure how this ends up becoming wastage, but maybe I'll end up finding out and adding it into the editing of the video later on. And so once you have your 14-desmethyl lanosterol, you add this to an NADPH molecule just as you did with the last reaction, as well as another O2 molecule, and this will turn into zymosterol. Again, there are no enzymes that catalyze this particular reaction, and there is only 2 CO2 molecules that end up coming out as wastage of this reaction. But once you get zymosterol, that's when it sort of gets a bit more easy to understand, because you can either go to zymostenol, and that is where you use 24 dehydrocholesterol reductase, which is an enzyme to get rid of the tail double bond. But you can also not do that step yet and do that step later on and get a molecule which I don't actually know the name. I couldn't actually find the name when I was doing my research, and I also don't know what enzyme catalyzes that reaction, unfortunately. But you end up getting a molecule that looks somewhat similar to cholesterol, where the double bond in the middle ends up being moved slightly down. And at this point, I'm not going to sort of explain all the different possible pathways. I'll get an image up on the screen which explains it very, very easily, and I'll edit this video so that you can sort of look and absorb that image for at least 10-20 seconds to get a grip and understand what this sort of whole thing is actually going on. But I'm going to go through a specific certain pathway. So we've already been through lanosterol, 14-desmethyl lanosterol, zymosterol, and then 24 reductase. We're going to do this pathway where you go to zymostenol, and after you get zymostenol, you then get lanosterol. And again, I'm annoyed because I forgot to put the enzyme names down on that I wrote down the biosynthesis on. But the step where you get zymostenol to lanosterol is similar to the step we talked about earlier, where you sort of just move the double bond from one place to another inside of the circles. And after lanosterol, you end up getting 7-dehydrocholesterol, and it actually has the name cholesterol in it, so this is the last step inside of this biosynthesis of cholesterol, which is, it's actually taken not as much time as I thought it would, which is great, but maybe with the editing and stuff, it'll end up having to put a few more images on screen and cutting out the video or the audio so that you can sort of absorb and understand it just that little bit better. But I also thought to put the enzyme name down in my notes for that reaction from lanosterol to 7-dehydrocholesterol, and what happens in this reaction is that it adds another double bond almost opposite to the other double bond that's already in that circle, and that's the actual place where the double bond needs to be inside of cholesterol. And then the reaction from 7-dehydrocholesterol to cholesterol removes the double bond that doesn't actually need to be there. And again, I forgot to put the enzyme down in my notes, which is very, very annoying, but hopefully I'll be able to sort of write it down on the screen and you can maybe search it up or whatever. So yeah, that is the complete biosynthesis of cholesterol. It's so like weird. It's quite a weird rabbit hole kind of thing. If you're watching this video still, this whole scientific aspect of our bodies, it's quite insane to think that all of these 50 maybe chemical reactions, not that I ended up being able to go through all 50 chemical reactions in this video, but all sort of the 20 that I went through, all of these are happening in your body maybe like incredibly quickly. And then, as I said before, once you get cholesterol, I'm pretty sure that ends up controlling the step that we ended up talking about earlier in the video. And cholesterol can be used for so many different things. It's insane. It ends up being used for bile acids to actually be able to digest your food. Ends up being used in like steroid hormones, testosterone, estrogen, progesterone, all that stuff, cortisol. But one problem with society nowadays is that it has demonized cholesterol quite a bit. And I think I read a book called The Optimum Nutrition Bible, and it said that the reason that cholesterol is demonized is because they did an experiment on rabbits where they gave it cholesterol, and the rabbits ended up getting quite a lot of like fatty deposits in their arteries. And that's because rabbits obviously don't digest cholesterol because it's not in their diets, and maybe they don't need it, I guess. I'm not quite sure how that works scientifically. Maybe they just produce it, but they don't actually sort of need it in their diet. Maybe that was what it was, but that's the reason that cholesterol is quite demonized. And it's quite interesting because cholesterol itself isn't the problem. Cholesterol is an incredibly healthy thing, but because cholesterol is fat soluble, it's not water soluble. This means that it can't be transported in the blood, as blood plasma contains things that are water soluble, as blood plasma is mostly water, to be honest. And so cholesterol has to be transported around the body to be used in its multiple functions by things called lipoproteins. And lipoproteins are fats and proteins, and the protein layer is the outside layer, so it can actually sort of be soluble in water. But the problem is that there are many different types of lipoproteins which carry cholesterol, and it's the type of lipoprotein which is the problem. HDL, high density lipoprotein cholesterol, and that would HDLC, high density lipoprotein cholesterol, that is a perfectly healthy way for cholesterol to be transported throughout the body. But there are other lipoproteins such as LDLC and VLDLC, low density lipoprotein cholesterol and very low density lipoprotein cholesterol. These are the big real killers in a way. These are the things that get clogged in the arteries and mean that cholesterol can't be transported, and it means that blood flow is severely hindered and stopped quite a lot. That's the problem. Cholesterol does not change. There aren't two different types of cholesterol. There's no good or bad cholesterol. It's the lipoprotein that's the problem. And I'm not quite sure how one can stop the bad lipoproteins being produced, but I assume it's something to do with maybe not eating sugar or not eating trans fats, if that makes any sense to you. So yeah, I really hope you enjoyed this kind of explanation on the biosynthesis of cholesterol. I know, despite the fact that I've been recording for nearly 2 hours now in the woods and my throat is absolutely dying, I thoroughly enjoyed just letting my chemical brain and scientific brain sort of spurt words out at you to the camera. And if they are wrong, if I did mention something wrong, please do tell me in the comments, because I don't want to spread misinformation on the internet. That would not be something that I want to do whatsoever. So yeah, I really hope you enjoyed, and there is a scientific Discord community which will be in the link in the description, and I highly recommend joining that if you want to sort of learn more about science and just be with people that are like you and have similar mindsets to you in general. So yeah, I hope you enjoyed, and I recommend subscribing because I love this community of young tribal people who are willing to improve themselves so much more than they already have. And good luck.